Hey class, this is Hugh again. I'm just doing a little note on DNA replication theory and on DNA transcription to RNA. So I'm just going to take a short piece of DNA. The order, remember, they're based on the nucleotides. In this case, adenine, cysteine, thymine, guanine, adenine, cysteine, guanine, and then it just goes on. It could be anything. Any of the four could be here. I'll put in a thymine, why not? And maybe then two A's. And then that'll be it for now. Okay. This end is a 5' end. This end is a 3' end, and there's a strand orientation. And you should have that from the previous note on DNA. Although you can have any code you want along here, and the code opposite side, it would run in the opposite direction. So if this is a 5' end here, this end would be 3', and going back this way to 5' would certainly be T, T, A, C, G, G, T, C, A, G, T, G, T. When DNA replicates, it is a complex process, and there is an assignment on that. But in the theory of it, this is the theory, is that the two strands separate first. So I have to have the two strands with their orientation. And here, T, G, T, G, A, C, T. Like that. One of the strands can be built as it is, because the copying machinery of the DNA polymerase enzyme reads from 3' to 5' and builds 5' to 3'. So let's suppose I have, this is my DNA polymerase. It moves from 3' to 5'. And as it does, it pops in the correct nucleotide. So if it sees an A, it pops in a T, and so on. The strand it builds will be, by definition, 5' to 3' as it gets through that. That's my dog. Padawan. Thank you. The other strand cannot be built in the same direction. So for that, another DNA polymerase has to grip on here and work its way backwards. And that's what the terms in your assignment, this would be called the leading strand. The one that's simply built as it needs to be built. And this is the lagging strand. It's built in little pieces, all going backwards. And so, same idea though, the enzyme works in the same way. So A. And you get that. And that would be the 3' end, 5' end. So DNA polymerase reads 3 to 5, it builds 5 to 3, no matter which strand it's working on. And these two now should be identical copies of the first, and that's the theory of DNA replication. You need to do a lot more in terms of doing the assignment, in terms of figuring out all the processes and how it works, but that's online for you. Today's topic mainly, though, is called transcription. Transcription is the changing of DNA codes into RNA codes. And the DNA piece is read. In a similar way, I'm going to use the top strand here and just replicate it. The enzyme that does this also runs in the 3 to 5 direction. And so In this case, the purpose is not to make another copy of the DNA, but rather to grab the information off of there in the form of this RNA molecule. Okay? So the enzyme that does it is called RNA polymerase. So it's the enzyme. I'll use a little blue dot like that. RNA polymerase starts somewhere on the DNA, and it's more complicated. We'll get into all those details later. But it starts on the DNA, reads it in the 3' to 5' direction, so it builds in the 5' to 3' direction for RNA. As it does so, it makes a copy of RNA based on the codes it reads here. So when it sees an A, right off the bat, we have to remember A does not have the T equivalent in RNA is U. So that would be a U. C would make G. A would make T. G. A. C. There's another A, so it's going to be a U. G. G. C. A. U. U. And this now is a piece of RNA, 5', 3', and this RNA has been transcribed. So the process of transcribing is using RNA polymerase to read one of the two strands of DNA, one of the two double strands, reading one of those and converting it into a message of RNA. If you notice, a very important thing is the RNA message here is exactly the same as the DNA of the other strand, except wherever you see a T, there's now a U. So this was TGTGA, UG, oops. Ooh, I made a mistake. Ooh, see? I made a mistake right there. Okay. That's how you catch your mistakes. So that's good. So it would be UGU, GACU, so GACT, GGCAUU. Transcription. Transcription then is followed later on. We'll worry about the RNA is converted into amino acids in a protein. And that's called translation. That's for a later date. So transcription, DNA is read, RNA is built. That's all.